Imagine a bacterial cell that sacrifices itself to protect its neighbors from viral takeover. This is the elegant strategy known as abortive infection. Abortive infection systems are a bacterial defense that sacrifices an infected cell to stop phage replication. When viral DNA enters the cytoplasm, host sensors detect foreign nucleic acids and trigger a rapid response. The response activates a cascade that shuts down essential cellular processes, preventing virus assembly. This self-destruct mechanism is irreversible. The cell lyses or ceases division within minutes of infection. Surviving neighboring cells inherit a protective state, often mediated by signaling molecules that prime defenses. The net effect is population-level immunity. The colony tolerates phage presence without widespread lyses. Abi systems are encoded on plasmids or chromosomes and can be transferred between strains by horizontal gene transfer. Their evolution is driven by the arms race with phages, leading to diverse molecular architectures. Key components include toxin-antitoxin modules that trigger cell death upon detection of viral DNA. Understanding Abi mechanisms informs synthetic biology, where engineered abortive responses can protect industrial microbial cultures. Pause 0.8. The type I restriction-modification complex patrols the bacterial cytoplasm for foreign DNA. It consists of a methyltransferase that marks host sequences and a restriction endonuclease that scans for unmethylated motifs. When an incoming phage genome enters, the complex recognizes its unmethylated recognition sites. The restriction enzyme then cleaves the viral DNA at defined positions, generating double-strand breaks. These breaks trigger a cascade that activates a programmed cell death pathway in the host. Activation involves rapid depolarization of the plasma membrane, disrupting ion gradients. The loss of membrane potential halts ATP synthesis and stalls essential metabolic processes. Consequently, the infected bacterium sacrifices itself to prevent further phage replication. This abortive infection strategy protects the bacterial population by sacrificing a few cells. The remaining uninfected bacteria survive and continue to propagate the species. Pause 0.8. CRISPR Cas systems act like a bacterial memory bank for viral DNA. When a phage injects its genome, the Cas1-Cas2 complex scans for foreign sequences. Short fragments of the invading DNA are excised and integrated as new spacers into the CRISPR array. These spacers become templates for transcribed cRNAs that guide the surveillance complex to complementary targets. The Cas9 or Cas12 nucleus, bound to the cRNA, searches the cell for matching viral DNA. Upon recognition, a double-strand break is introduced at the target site, disabling the phage genome. If the break cannot be repaired, the bacterial cell initiates an abortive infection program to prevent further replication. This program death sacrifices the infected bacterium, but preserves the overall population from viral spread. The process is highly specific. Only sequences that match the spacer are cleaved, sparing host DNA. Thus, CRISPR-Cas provides a precise, adaptive defense that turns viral invasion into a self-terminating event. Pause 0.8. The PSB system monitors the bacterial membrane for sudden changes. When a phage injects DNA, it disturbs the lipid bilayer integrity. This perturbation triggers the sensor protein PS-beta to change shape. PS-beta then signals the transcriptional regulator to upregulate PSB genes. The newly produced PSBB and PSBC proteins reinforce the membrane structure. If stress persists beyond a safe level, the system halts cell division. The halted state is called dormancy, preventing phage replication. Alternatively, the cell may undergo lyses to release toxic factors. These outcomes are collectively termed abortive infection. Thus, the PSB response protects bacterial populations at the cost of individual cells. The threshold for abortive activation is sensed by a feedback loop involving PSBF that shuts down essential metabolic genes when membrane tension rises. This coordinated shutdown ensures that surviving bacteria can recover once phage pressure subsides. Pause 0.8. In many bacteria, a toxin and its antitoxin form a tightly bound pair that keeps the toxin inactive. The antitoxin is produced at a steady rate and binds the toxin's active site. When a bacteriophage injects DNA, Host sensors trigger rapid degradation of the antitoxin protein. This frees the toxin to interact with essential cellular targets such as ribosomes or replication enzymes. One common target is the ribosomal decoding center, where the toxin blocks mRNA movement. As translation stalls, proteins required for phage assembly cannot be made. The cell's metabolic activity drops sharply, and energy stores are depleted. This loss of viability leads to a form of programmed death that stops further viral replication. By sacrificing itself, the bacterium prevents the spread of infection to neighboring cells. Thus, toxin-antitoxin modules act as an abortive infection system that protects bacterial populations. Pause 0.8. Abortive infection causes a single bacterium to sacrifice itself. When a phage invades, this sulfurose destruct mechanism prevents the phage from replicating and spreading. Over evolutionary time, bacteria that can trigger abortive infection survive more often. Consequently, genes encoding ABI systems become widespread across diverse bacterial lineages. Phages counter this by evolving mutations that bypass or inhibit the host's abortive response. The resulting arms race drives rapid genetic diversification in both partners. Phylogenetic analyses reveal clusters of ABI genes that correlate with specific phage families. These patterns suggest co-Euro's evolutionary hotspots where bacteria and phages repeatedly interact. In biotechnology, engineered bacteria can be equipped with optimized ABI modules to block harmful phages in industrial fermentations. Such designs also enhance the safety profile of phage therapy by limiting unintended bacterial death. Pause 0.8. Abortive infection is a bacterial strategy where infected cells trigger sulfurose destruction to stop phage spread. Mechanisms include RM cleavage, CRISPR Cas targeting, PSB stress response, and toxin-antitoxin activation. These systems shape bacterial evolution and offer insights for phage therapy.